Today's lesson, Spirochetes. In this lesson, we are going to tackle syphilis, Lyme disease, and Borrelia miyamotoi and their introduction and lab diagnosis. Here are the reporters: Bloomchen Agramon, Mary Angela Dairo, Joshua Flores, Cristina Asuncion, Chad Muyot, Kate Ramos, and Maurice Victolero. So we are quite familiar in spirochetes, right? Kasi yung mga previous lesson in bacteriology. But let us recall pa din what spirochetes are. So spirochetes are long, slender, helically coiled bacteria containing axial filaments or periplasmic flagella, which wind around the bacterial cell wall and are enclosed by an outer sheet. These gram-negative microaerophilic bacteria exhibit a characteristic corkscrew flexion or motility. For a more detailed discussion about spirochetes, let us move on to our reporters.
So good day everyone, I am Chadren Simuyat and I will be discussing syphilis. So syphilis is the most commonly acquired spirochete disease in the United States. The causative agent of syphilis is Treponema pallidum, subspecies pallidum, which is a member of the family Spirochetaceae. So organisms in this family have no natural reservoir in the environment and must multiply within a living host. Pathogenic treponemes are rapidly destroyed by heat, cold, and drying, so they are almost always spread by direct contact. So sexual transmission is the primary mode of dissemination, which occurs through contact of abraded skin or mucous membranes with an open tissue. Other potential means of transmission include parenteral exposure through contaminated needles or blood, but this is extremely rare. Untreated syphilis can progress through 4 stages, which are the primary, secondary, latent, and the tertiary stages. So once contact has been made with a susceptible skin site, endothelial cell thickening occurs with aggregation of lymphocytes, plasma cells, and macrophages. The initial lesion, called a chancre, develops between 10 and 90 days after infection. So about 25% of patients who are untreated in the primary stage progress through the secondary stage, in which systemic dissemination of the organism occurs. The latent stage follows the disappearance of secondary syphilis. So the latent stage is characterized by a lack of clinical symptoms. It is arbitrarily divided into early latent and late latent, in which the primary infection has occurred more than 1 year previously. Patients are non-infectious at this time with the exception of pregnant women, who can pass the disease onto the fetus even if they exhibit no symptoms. So about 1/3 of the individuals who remain untreated develop tertiary syphilis. So this stage occurs most often between 10 and 30 years following the secondary stage. And tertiary syphilis has 3 major manifestations: syphilis, cardiovascular disease, and neurosyphilis. Now let's move on to the laboratory diagnosis of syphilis. So direct detection of spirochetes can be accomplished by their dark field microscopy or fluorescent antibody testing. The performance of either test requires that the patient have active lesions. So primary and secondary syphilis can be diagnosed by demonstrating the presence of Treponema pallidum in exudates from skin lesions. So in dark field microscopy, a dark field condenser is used to keep all incidental light out of the field except for that captured by the organisms themselves. So it is essential to have a good specimen in the form of serous fluid from a lesion. The serous fluid is usually obtained by cleaning the lesion with sterile saline and rubbing it with clean gauze. Pathogenic treponemes are identified on the basis of characteristic corkscrew morphology and flexing motility. So because observation of motility is key to identification, specimens must be examined as quickly as possible before they dry out.
So, I'm Kate Faye R. Ramos and I'll be discussing the fluorescent antibody testing. So for the fluorescent antibody testing, it is a sensitive and highly specific alternative to dark field microscopy. And it can also be performed either the direct or the indirect method. So in the direct method, it uses a fluorescent labeled antibody conjugate to T palladium, or an indirect method using antibody specific for T palladium and a second labeled anti-immunoglobulin antibody. And with the use of the monoclonal antibody, it has made fluorescent antibody testing very sensitive and specific. So next is the serological testing. It can be classified into non-treponemal and the treponemal test, but I will discuss first the non-treponemal test. It is used to screen for syphilis because of their high sensitivity and ease of performance. It also determines the presence of an antibody that forms against a cardiolipin, which is the lipid material released from the damaged cells. The reagent can be found in the sera of the patients with syphilis and several other disease states. The term reagent as it applies to syphilis should not be confused with the same word that was originally used to describe the IgE. Fortunately, the term reagent in reference to IgE is rarely used today. There are two types of tests that is widely used in the non-treponemal test. These are the VDRL, which is the Venereal Disease Research Laboratory test, and the RPR test, which is the Rapid Plasma Reagent test. These tests are based on the flocculation reaction in which the patient antibody complexes within the cardiolipin antigen. The flocculation, this is a specific type of precipitation that occurs over a narrow range of antigen concentrations. The VDRL test is both a qualitative and quantitative slide flocculation test for the serum that includes a modification for use on the spinal fluid. The serum specimen to be tested are heated at 56 degrees Celsius for 30 minutes to inactivate the complement, and the test must be performed at room temperature within the range of 23 degrees Celsius to 29 degrees Celsius because the temperature may affect the results. All sera with reactive or weakly reactive results must be tested using the quantitative slide test, in which two-fold dilutions of serum ranging from 1:2 to 1:32 are initially used, and sera yielding the positive results at the 1:32 dilution are titered further. For the rapid plasma reagent test, or the RPR test, it is a modified VDRL test involving the macroscopic agglutination. The antigen is similar to the VDRL antigen with the addition of the ethylene diamine tetraacetic acid dimerosal, the choline chloride, which it stabilizes the antigen and inactivate complement so that the serum does not have to be heat inactivated before use. The cards are read under a high intensity light source, so if the flocculation is evident, the test is positive. All reactive tests should be confirmed by retesting using doubling dilutions in a quantitative procedure. So the RPR test appears to be more sensitive than the VDRL in the primary syphilis. So this is the RPR test result in an 18mm circle on a plastic coated disposable card. So the well 1 reactive control showing large clumps, and the well 2, which is the weakly reactive control, showing small clumps, and the well 3 is the non-reactive control showing slight roughness and a tail upon swirling. And the well 6 to 10 shows a result of a serially diluted sample of patient serum with a titer of 8.
Good day everyone, I am Mary Angela S. Dyer, here to present the treponemal test under serological test. Treponemal test detects antibody directed against the T palladium organisms or against specific treponemal antigens. Treponemal tests usually become positive before non-treponemal tests, although patients with early primary syphilis may be non-reactive. And secondary and latent syphilis tests are usually 100% reactive. Two types of manual treponemal tests are the indirect fluorescent treponemal antibody absorption test and the agglutination test. Because these tests are highly specific for syphilis, they have been used to confirm positive non-treponemal test results. The fluorescent treponemal antibody absorption is one of the earliest confirmatory tests. In this test, a dilution of heat and inactivated patient serum is incubated with a solvent consisting of an extract of non-pathogenic treponins, which removes antibodies that cross-react with treponins other than T. pallidum. Diluted patient samples and controls are applied to individuals' wells on a test slides fixed with nickel strain of T. pallidum. Following a 30 minutes incubation at 37 degrees Celsius, the slides are washed and air-dried, and antibody conjugate is added to each well. Slides are re-incubated as before and washed to remove excess conjugate. Mounting medium is applied and cover slips are placed on the slides. They are then examined under fluorescence microscope. The particle agglutination test originally used ships RBC coated with T. pallidum antigen and were referred to as microhemagglutination assay for T. pallidum. Current particle agglutination tests for T. pallidum, such as the Seroja T. pallidum particle agglutination test, use colored gelatin particle coated with treponemal antigens and are more sensitive in detecting primary syphilis. Patient serum or plasma is diluted in microtiter plates and incubated with either T. pallidum sensitized gel particles or unsensitized gel particles as a control. Presence of T. pallidum antibodies is indicated by agglutinations of sensitized gel particles, which form a lattice-like structure that spreads to produce a smooth mat covering the surface of the well. If a sample is negative for the antibody, the gel particles settle to the bottom of the well and form a compact button. Now, let us talk about the special diagnostic areas, which are the congenital syphilis and the cerebrospinal fluid. Non-treponemal tests for congenital syphilis performed on cord blood or neonatal serum detects the IgG class of antibody in addition to IgM. It is difficult to differentiate passively transferred IgG maternal antibodies from those produced by the neonate, so there are problems in establishing a definitive diagnosis. Late maternal infection may result in a non-reactive test because of low levels of fetal antibody. Additionally, testing the infant's spinal fluid for the presence of treponins often lacks sensitivity. Non-treponemal titers in the infant that are higher than those in the mother may be a good indicator of congenital disease, but this not always occur. Currently, it is recommended that in high-risk populations, non-treponemal tests be performed on both the mother and infant birth, regardless of previously negative maternal tests. Because symptoms are not always present at birth, if congenital syphilis is suspected because of maternal history, tests should be repeated on infant serum within a few weeks. If infection is present in the infant, the titer will remain the same or will increase. The Western blot test is recommended to confirm the congenital syphilis.
Good day everyone, I'm Joshua Flores and I'll be discussing now the cerebrospinal fluid under the special diagnostic areas. So CSF is used to determine whether the treponins have invaded the central nervous system. Such testing is usually more reliable if the central nervous system symptoms are present. The VDRL test and some of the newer ELISA tests are the only ones routinely used for the testing of the spinal fluid. For VDRL spinal fluid testing, the antigen volume used is less than the serum test and is at a different concentration. The test is read microscopically as in the VDRL serum test. If a test is reactive, twofold dilutions are made and retested following the same protocol. A positive VDRL test on a spinal fluid is a diagnostic of neurosyphilis because false positives are extremely rare. However, sensitivity is lacking because samples from less than 70% of patients with active neurosyphilis give positive results. If a negative test is obtained, other indicators such as increased lymphocyte count and elevated total protein with a measurement at 45 milligrams per deciliter are used as a sign of the active disease. Okay, now moving on to the next topic is Lyme disease. Let's have first a brief introduction or background about this disease before my groupmate discuss to you the laboratory diagnosis. So Lyme disease was first described in the United States in 1975 when an unusual large number of cases of juvenile arthritis appeared in a geographically clustered rural area found around Old Lyme, Connecticut, hence the disease name Lyme disease, in the summer and fall. In 1982, the agent was isolated and identified as a new spirochete. It was given the name Borrelia burgdorferi after Willy Burgdorfer, which was the first author in the original description. The clinical features of Lyme disease were soon recognized to extend beyond arthritis. It is now known to be a multi-system illness involving the skin, nervous system, heart, and joints. Lyme disease is the most common vector-borne disease in the United States. Over 36,307 confirmed or probable cases were reported back in 2013. The clinical hallmark of early infection is the rash known as the erythema migrans, which you can see on the right side of your screen, the second picture. And this appears between 2 days and 2 weeks after a tick bite. The erythema migrans begins as a small papule where the bite occurred, then rapidly expands to form a large ring-like erythema and often a central area that exhibits partial clearing. The erythema migrans usually continues to expand for over a week. Even if untreated, it gradually fades within 3 to 4 weeks. Early dissemination occurs via the bloodstream in days to weeks following the erythema migrans rush. The skin, nervous system, heart, or joints may be affected, and approximately 10% to 15% of patients will display multiple skin lesions. Migratory pain often occurs in the joints, tendons, muscles, and bones. If treatment is not obtained, neurological or cardiac involvement develops in about 15% of patients within 4 to 6 weeks after the onset of the infection. And the most prevalent neurological sign is facial palsy, a peripheral neuritis that usually involves one side of the face. Pain and weakness occur in the limb that was bitten. Some patients develop sleep disturbances, mild chronic confusional states, or difficulty with memory and intellectual functioning. An aseptic meningitis can also be seen.
The IFA was the first test used to evaluate the antibody response in Lyme disease, followed by various forms of EIAs shortly thereafter. The IFA assay is fairly easy to develop, which is why it is usually the first assay on the market in many infectious disease arenas. Basically, doubling dilutions of patient serum are incubated with commercially prepared microscope slides coated with antigen from whole or processed spirochetes. Following a wash step to remove unbound material, an anti-human globulin with a fluorescent tag attached is added and reacts with any specific antibody bound to the spirochetes on the slide. After a second wash step, the slide is viewed under a fluorescent microscope. Typically, a test result is only considered positive if a titer of 1:256 or higher is obtained, although this varies between manufacturers. As previously mentioned, specimens obtained in the first few weeks are usually negative because the level of antibody present is below the detection limit of this and other assays. As might be expected, other closely related organisms such as Spirochetes associated with relapsing fever, Lidenticola, and others associated with periodontal disease, and T. pallidum for syphilis may cross-react and cause biological false positive results. Autoimmune connective tissue diseases such as rheumatoid arthritis and SLE can also produce false positives in the IFA assay for Lyme disease and FDA assay for syphilis. An astute technologist can recognize a false positive by the beaded fluorescent pattern it produces. Reading of fluorescent patterns tends to be very subjective and requires highly trained individuals. However, if performed correctly by experienced personnel, the test can provide sensitive and accurate results. This test is best suited for low-volume testing. EAA testing is quick, reproducible, mass-objective, relatively inexpensive, and lends itself with automation in high-volume testing. Antigen preparations used in the assay include pure sonicates of the organism, purified proteins, synthetic proteins, and recombinant proteins. The manufacturer's selected antigen is then coated onto 96-vial microtiter plates or strips by various proprietary methods. Patient sera is added and allowed to incubate with the antigen. After a washing step, anti-human globulin conjugated with an enzyme tag such as alkaline phosphatase is added to each vial. Adding specific substrate produces a color change. Plates are read in a spectrophotometer and the antibody is quantitated based on color intensity. EIAs provide objective results, and the titer is based on a continuum range rather than serial dilutions of patient sera. Thus, a more accurate measurement of the specific antibody is possible. Similar to the IFA, drawbacks of EIA include a lack of sensitivity during the early stages of Lyme disease and specificity problems. The sensitivity for early serum specimens has been reported to be anywhere from 58% to 92%. The differences in using various antigens is usually manifested in trade-offs between increasing sensitivity at the expense of specificity versus increasing specificity at the expense of sensitivity. Unfortunately, the technical adaptation of the EIA have not resulted in high enough sensitivity and specificity to replace the two-tier method of testing. The specificity issues are generally similar to the IFA assay. However, there are no clues or beaded pattern that a particular sample may be a false positive. As with IFA, false positives occur with syphilis and other treponema diseases such as yaws and periodontal disease, as well as relapsing fever and leptospirosis. If serum is absorbed to decrease cross-reactivity, this also decreases specific Lyme antibody titers. Additionally, patients with infectious mononucleosis, Rocky Mountain, spotted fever, and other autoimmune diseases also have been known to be positive with EIA. Lyme disease patients do not test positive with RPR. So, this may be helpful if syphilis is in a differential diagnosis.
Hello everyone, I am Maurice Victolero and we will be talking about the Western blot and the polymerase chain reaction as the laboratory test for the diagnosis of Lyme disease. Of course, when talking about laboratory diagnosis of Lyme disease, Western blot will be in the list kasi siya ay second test in the CDC recommended two-tier testing scheme for Lyme disease. So, immunoblotting, also known as Western blotting, ay confirmatory test siya for samples na initially nag-test positive or equivocal by enzyme immunoassay or immunofluorescence assay. If yung patient had symptoms for more than 4 weeks na, they are not advised to be tested for IgM antibodies. Kasi instead, a positive result ng IgG immunoblot indicates serological evidence ng Lyme disease. So, the Lyme disease immunoblot is a complex test that consists of electrophoresis of Borrelia antigens in an acrylamide gel and then transfer of the resulting pattern to a nitrocellular paper. The nitrocellular antigen strips are provided na sa test kit and are reacted with patient serum. They are then developed with anti-human globulin to which an enzyme label is attached. The further incubation with the enzyme substrate allows for visualization of any antibody that has bound to a particular antigen. Tapos, yung reactivity niya is scored and interpreted. So, paano siya i-interpret? So, ganito. So, meron tayong 10 proteins used in the CDC recommended interpretation of this test based sa molecular weights nila. For a result to be considered positive for the presence of a specific IgM antibody, 2 of the following bands must be present. Ito yung 23 OSPC, 39 and 41 flagellin KDA, and IgG immunoblot is considered positive naman if 5 out of 10 bands ay magpa-positive. So, yung 10 is yung makikita natin sa figure 2111, which is the 18, 23, 28, 30, 31, 41, 45, 58, 66, and 93 KDA. Now, the PCR test naman for Lyme disease. So, the PCR test has found a specific use in certain scenarios. Although only a few organisms need to be present for detection under optimal conditions, the number of spirochetes in infected tissues and body fluids is low, making specimen collection, transport, and preparation of DNA critical to the accuracy of the test results. Pero there are already several probes for target DNA that is present only in the strains of Borrelia burgdorferi na nagagamit na natin sa PCR testing. So, familiar naman tayo sa PCR test, diba? So, ganitong procedure niya. So, first, extracting DNA from the patient sample, followed by amplification using specific primers, DNA polymerase, and nucleotides. Once a sufficient amount of the unknown DNA is made, this is combined with the known DNA probe to see if hybridization takes place. So, the single-stranded Borrelia DNA probe will bind only to the extra complementary strand, thus positively identifying the presence of the organism's DNA in the patient sample. So, mas specific siya kaysa yung antibodies ang ating gamitin kasi mayroong little cross-reactivity. So, mayroong recent PCR studies na nagpapakita ng specificity ranging from 93 to 100%. However, sensitivity remains problematic. In a series of studies, the median sensitivity of PCR on skin biopsies 69%, blood components 14%, CSF 38%, and synovial fluid 78%. Pero, yung range of sensitivities in any one type of specimen is quite large naman, suggesting that testing remains to be standardized. Furthermore, it would be hard to clinically justify a skin biopsy for PCR as a diagnostic method for an EM rash in most cases. So, however, in difficult diagnostic neurological and arthritic cases, PCR on CSF and synovial fluid is often employed. Pero, ang sabi, PCR for Borrelia still has limited availability.
Borrelia miyamotoi. B. miyamotoi is closer phylogenetically to the relapsing fever Borrelia group than it is to B. burgdorferi. However, similar to B. burgdorferi, it has at least one mammalian reservoir host, like B. burgdorferi. The white-footed mouse is preferred host in the eastern United States. The infection prevalence of questing nymphs in endemic areas is quite different from B. burgdorferi. It is 20% to 50% in B. burgdorferi and only 1% to 5% for B. miyamotoi. Unlike B. burgdorferi, but similar to other relapsing fever Borrelia species, vertical transmission from the female adult tick to its offspring occurs in addition to the horizontal transmission from the reservoir host to the tick. This transmission to offspring has possible implications for transmission to humans because the extremely small tick larvae are potentially infectious.
Good day, everyone. My name is Christina Asuncion, and I am here to discuss about the laboratory diagnosis of Borrelia miyamotoi infection. First is direct detection by culture or PCR. The laboratory diagnosis of P. miyamotoi infection is still in its infancy. At the time of this writing, all diagnostics are basically research and investigational only, and there are no commercial FDA-approved kits. Although B. burgdorferi can be cultured, it is not useful as a diagnostic tool because it is a low-yield procedure. Attempts to culture B. miyamotoi in media have not been successful to date. However, if improved culture techniques are developed, it may be a viable diagnostic option because of the high-density bacteremia. It is also possible that PCR testing of blood may become a feasible diagnostic method in the future. The diagnosis of Borrelia miyamotoi is most often made by clinical examination combined with evidence of tick bite or known or presumed exposure to ticks in endemic areas. Amplification of B. miyamotoi genomic DNA from blood, fluids, or tissues confirm the diagnosis. Polymerase chain reaction or PCR analysis is used to confirm acute B. miyamotoi infection. However, if a patient does not have access to that test, conventional laboratory assays may be informative. This test could show low white blood cell count, elevated liver function test, low platelets, and protein in urine. The specific Borrelia miyamotoi test can be extremely helpful in a case where the patient has no rash, a negative Western blot, but presents with flu-like symptoms. It has been reported that occasionally, a patient with Borrelia miyamotoi will test falsely positive on the Lyme C6 peptide ELISA. There is no spinal fluid antibody assay for Borrelia miyamotoi. The second one is the antibody detection. The Russian study, which first found the disease associated with P. miyamotoi infection, demonstrated, not surprisingly, that these patients were positive in the Lyme antibody assays. Thus, it is possible that some of the diagnosed Lyme patients could actually be B. miyamotoi patients instead. The 2013 U.S. study that confirmed B. miyamotoi infection used an ELISA assay, which detected antibodies against GLP2 proteins, which are made from B. hermsii. These antibodies are specific for the relapsing fever Borrelia and not B. burgdorferi. Seroconversion is assessed using the glycerophosphodiesterase or GLP2 enzyme immunoassay. IgM antibodies have been found to be reactive between 11 and 20 days after disease onset, and IgG antibodies reactive 21 to 50 days after disease onset. The GLP2 protein is present among all relapsing fever Spirochetes but absent in burgdorferi. B. miyamotoi infections are treated with a 2 to 4 week course of the antibiotics doxycycline, amoxicillin, and ceftriaxone have also been successfully used.
That is all. Thank you for listening.